Now that we understand the basic process of mitosis, we need to take a closer look at the mitotic spindle because this structure is incredibly important in precisely separating the duplicated chromosomes during mitosis. So the mitotic spindle is made out of essentially three parts. I've got it numbered here. So first of all, you've got the centrosomes. Number two, you've got the asters, and then lastly, you've got the spindle microtubules. Let's talk about the centrosome first. This should be a review. So remember that the centrosome is described as the microtubule organizing center of the cell. And this is where microtubules that are used to separate the sister chromatids are actually constructed. In animal cells, the centrosomes, right, these structures are also typically going to have those centrioles. The little pasta-shaped microtubule structures are always perpendicular to each other inside of the centrosome. And again, remember, we don't know what the function of the centrioles actually is. We think that maybe it's some sort of evolutionary relic that has remained inside of the animal cells. Also keep in mind that the centrosomes, typically a cell only has one of them, but in preparation for division during interphase, the centrosome gets replicated so that the cell ends up with two of them, one for each pole of the cell to pull the chromosomes to opposite poles. Now, the other two parts of the mitotic spindle include microtubule structures. So the asters, this is not something we've taken a look at yet, but the asters are really short microtubule structures that point away from the center of the cell. So they extend from the centrosome, but point sort of outward to the edge of the cell, sort of anchoring the centrosome in place. The spindle microtubules are, again, microtubules, but they're extended in the opposite direction. They extend from the centrosome towards the center of the cell. And it's these spindle microtubules that during prometaphase start to attach to the duplicated chromosomes and play that sort of tug of war from opposite ends of the cell to line all of the chromosomes up at that metaphase plate. Now, something that we haven't covered yet is how exactly those spindle microtubules actually attach to the duplicated chromosomes. So let's take a quick look. If we've got our, let's say, our single chromosome, right? In S phase, that single chromosome gets duplicated. So now we still have one chromosome, but it's a duplicated chromosome made out of those two sister chromatids. Remember that narrow waist here of the duplicated chromosome, this is called the centromere. And at that centromere, there's a protein structure that gets added to both of the sister chromatids. And that protein structure is called a kinetochore. Spelled kind of funny, kinetochore. And it's this kinetochore that acts kind of like a little landing pad for the microtubules. So microtubule from this pole of the cell will attach here. Another microtubule from the other pole will attach there. Another one, and another one. And this way, the opposite poles, as the microtubules attach to the kinetochores, play sort of a back and forth tug of war with those duplicated chromosomes. And so what that does is it helps to line up all of those duplicated chromosomes at that metaphase plate. And so again, you can see that here in the cartoon diagram, but also here in the electron micrograph. So right here, the structure at the centromere of the chromosome, that's going to be the kinetochore. You got another kinetochore here, and you can see all of the microtubules on either side that have attached to the sister chromatids via the kinetochore. Now, keep in mind that when you're looking, let's say in lab, for cells under the microscope that are in metaphase, cells in real life don't look this neat. This is a cartoon diagram where all of the chromosomes are nicely, neatly, perfectly lined up at the metaphase plate. Real life is a little more messy. So again, this is an example of part of a cell. The cell is going through metaphase. And yeah, you can imagine that all of the centromeres of the chromosomes are nicely lined up there in the middle at that imaginary metaphase plate. But then these chromosome arms, they're really long and they're going to dangle, on one side or another side of that metaphase plate. So it's not going to be as nice and neat as what you see in the cartoon diagrams. All right, so once all of those chromosomes are lined up at metaphase, the cell's ready to split them apart, split apart the sister chromatids in anaphase. Remember that the sister chromatids are held really tightly together using those cohesin proteins. And the cohesin proteins need to be cut. And so there's an enzyme that's called separase that helps to separate the sister chromatids. It essentially is like a pair of scissors, and it comes along and it cuts all of those cohesins, separating the two sister chromatids. As soon as they're separated, remember, they become full-fledged chromosomes, and they immediately start moving to opposite poles of the cell. They move with the help of those spindle microtubules. Now, the tricky part is there are two kinds of spindle microtubules. There are what we call kinetochore microtubules. These are the ones that extend from the centromere and actually contact a kinetochore, and they help to pull the chromosomes to opposite poles of the cell. During anaphase, these guys are going to shorten, right? As they shorten, chromosomes get pulled to the pole. There are also what we call non-kinetochore microtubules. If you look at this pair of microtubules right in the center of the cell here, you'll notice that they don't touch a chromosome. Instead, they sort of overlap each other, and they hold hands in the middle of the cell. And these guys, the non-kinetochore microtubules, actually lengthen as the cell goes through anaphase, and that ends up pushing the two poles of the cell further and further apart, helping to prepare the cell for cytokinesis. Eventually, the entire mitotic spindle or spindle apparatus is going to sort of be deconstructed or disassembled, and all the microtubules will be taken apart, and then you'll just have a centrosome that's left in each of the sister cells at the end of mitosis and cytokinesis. Now let's talk a little bit about cytokinesis, because this can happen a little bit differently depending on what kind of cell you're looking at. So in animal cells, I'm going to back up just a little bit. When we were looking at the different phases of mitosis here on the top, real images of cells, and then the cartoon diagrams on the bottom, these are all of animal cells going through cell division. So cytokinesis in an animal cell, we're looking for what is called a cleavage furrow, or this area within a cell that you can see that the cell is starting to pinch in. How cytokinesis happens in these animal cells is that they have this ring of proteins that can contract. And as the cell goes through cytokinesis, the ring of contractile proteins is going to contract and contract and contract until it literally pinches the cell in two. So it's kind of like, imagine, you're sitting in a pair of sweatpants, and the sweatpants have a drawstring. What would happen if you pulled that drawstring tighter and tighter and tighter until you pinched yourself in half? That's exactly what an animal cell does to go through cytokinesis. Plant cells, very different, right? Plant cells have a rigid cell wall. They can't just pinch themselves in half, right? The cell wall is not going to allow that. So instead, they need to build a new cell wall in between the two daughter nuclei. So maybe let's take a look at how that might happen. So if we've got, let's say, a cell, and I'm going to draw its plasma membrane here in green. And it's finishing up mitosis, so it's got the two daughter nuclei. Now remember that a cell wall for a plant cell is made out of cellulose, right? So let's draw this cell wall made out of cellulose and, other proteins and carbohydrates in red. So the cellulose that's used by the cell to build its cell wall, it's actually produced by the cell itself. So what it's going to do is it's going to make the cellulose on the inside of the cell using its endomembrane system. It's going to package it into these small vesicles, so they're membrane bound, and they have all this cellulose or cell wall material on the inside. Usually a cell will just send those vesicles to the plasma membrane, and there they'll fuse and they'll dump all the cellulose out to the outside of the cell, and you can build up your cell wall that way. But during cytokinesis, it's a little bit different. The cell instead will send all of those vesicles here to the middle of the cell, so in between the two daughter nuclei. And as more of those vesicles accumulate, what's going to happen is they start to fuse together And then they create this very large membrane-bound structure that's literally just chock full of this cellulose material. All right, and this whole entire structure then that's in the middle of the cell, this guy is called a cell plate. And eventually, as more and more of those vesicles fuse, you'll end up allowing that cell plate to fuse with the existing plasma membrane, and you'll have a nice continuity of the cell wall from the cell plate to the existing cell wall on the outside. And you'll have two completely separated plant cells. All right, so remember that if you're looking for a cell, evidence of cytokinesis in an animal cell, you're looking for the cleavage furrow, a pinched in region. If you're looking for evidence of cytokinesis in a plant cell, you're going to look instead for that cell plate, the structure sort of in the middle of the two daughter nuclei that have just formed or are finishing formation. To help you, because we've gone through a lot of examples of animal cells going through mitosis, there are slides and images in your textbook that will take you through mitosis in plant cells, just to give you an idea of what you would look for. And then I'll cover just this last slide. In telophase, right, telophase overlaps with cytokinesis. Here you can see the formation of the cell plate. It hasn't fused yet with the existing cell wall, but it's being built as mitosis is finishing to create those two daughter nuclei.